we present the mechanism of insulin secretion. Imagine you've just eaten a large meal. You've broken down your carbohydrates, and now your blood sugar, that is, your blood glucose levels, go up. The glucose molecules travel to the pancreas and end up in the fenestrated capillary network of one of the many islet of Langerhans within the pancreas. They exit the fenestrated capillaries and find themselves amongst the insulin-producing beta cells of these islet of Langerhans. The glucose molecules enter the beta cell via the GLUT2 transporter, a transporter also found in liver hepatocytes. In contrast, tissues like adipose or skeletal muscle possess GLUT4 transporters instead. The glucose is then quickly phosphorylated by the enzyme glucokinase into glucose 6-phosphate. The negatively charged phosphate group makes it more difficult for the glucose 6-phosphate to exit the cell, essentially trapping it within the beta cell. The glucose 6-phosphate is then metabolized through glycolysis and the citric acid cycle, eventually giving us a number of ATP molecules. These energetic ATPs bind to and close the ATP-sensitive potassium channels of the cell membrane. Because there is normally a higher concentration of potassium within cells, potassium ions try to leave the beta cell, but less of them are able to, and so they remain inside and depolarize the cell. This depolarization triggers the opening of voltage-gated calcium channels. Now, calcium ions, which are more abundant extracellularly, enter the cell. These calcium ions stimulate the fusion of insulin-containing vesicles with the plasma membrane, leading to the release of insulin outside the cell. The insulin molecules then diffuse into the fenestrated capillaries and find themselves in the bloodstream, ready to act on other tissues and decrease blood sugar levels. What we have just described is the first phase of insulin secretion. After several minutes, a second phase kicks in, involving the recruitment of more vesicles from a reserve pool within the cell, as well as the activation of more enzymes related to insulin production. It is not only glucose that affects insulin secretion, as we shall soon find out. First of all, amino acids and fatty acids can be metabolized and eventually also yield ATP molecules, increasing insulin secretion. Parasympathetic stimulation with the release of acetylcholine also stimulates insulin secretion. Another class of molecules, called incretins, which includes GLP-1 and GIP, increase insulin secretion as well. These incretins are released from special enteroendocrine cells of the intestinal epithelium. GLP-1, that is, glucagon-like peptide 1, is produced by L cells, whilst GIP, gastric inhibitory peptide, is produced by K cells. In response to a meal, these guys are released and travel through the bloodstream to reach the beta cell and increase insulin secretion. Indeed, drugs that are GLP-1 agonists, which act like our body's GLP-1, are used to stimulate insulin secretion in type 2 diabetics. Thank you for watching. From all of us here at All Night Med.